We're going to get this started as soon as we can find somebody with a mini DisplayPort do VGA adapter. Someone walked off with ours, and it was not Steve Rombaum. I know his was his. All right. Thank you. Don't forget to pick it up after the talk. Yeah. Yeah, they're going to see.
Wouldn't mind getting horizontal. It's on the side here. There we go. This is a new laptop. You're the first one to do that.
I'm gonna have to change your resolution.
Yeah, no, go for it. No. Oh yeah. Yeah, so it's scaled.
Yeah, now anybody else hear that? No, the mic's on.
Oh, there we go.
There we go.
Great. Thanks, man.
Okay, you're on.
Oh, wow.
Okay, we're ready to roll.
Great. Okay, everybody, we're going to start
Wait,
now.
I've just got to do a quick intro. We had two. I'm on the speaker selection committee. We had hundreds of submissions. Two of them were about biohacking, and when the other submitters heard the qualifications of these two folks here, they just bowed out. They're like, we can't come even close to their qualifications. So I'll let them introduce themselves, but they're, needless to say, Canadians, and they're going to talk about biohacking north of the 45th parallel. Yay, Canadians.
Okay, so thanks for the intro, and you're totally correct. Our credentials are absolutely fantastic. I studied in school for 14 years, and I wound up getting a 3-year film degree out of that. But at the same time, I also talked my way into being a visiting scholar at the MIT Media Lab, doing a stint at NASA Ames as well. And actually, that's when I got started in biohacking, switching over from computing and human-computer interaction and things like that over to biohacking. And now, I guess maybe I'm a poster person for the fact that, hey, you don't need to be a biologist to get involved in biohacking. And also, before I pass the mic to Kev, biohacking is the newest, coolest thing, and if you're not convinced of that, by the end of our talk, come chat with us later on and I can tell you more reasons why.
Cool. That's a very nice introduction. I'm Kevin. I'm a biologist, I guess, by trade. And my background in biohacking comes from the International Genetically Engineered Machine Competition, or IGEM, in case you guys have heard of that. But I spent 4 years getting a 4-year degree in biochemistry. So, yeah. And now I guess we co-founded the Montreal biohacking group called BricoBio. And currently I'm coming from Cork, Ireland, actually, because I'm working on a company at the SynBio Accelerator program over there. So that's my story.
Awesome. So yeah, we're going to just basically dive into it. It's not going to be super rigorous. It's not going to be super long. If anyone has any questions, feel free to interrupt. But, you know, maybe at the end would be better. But yeah, so on the screen now, this is our Twitter stuff. You can write that down for later to tweet at us or to us and all that kind of stuff. But the first thing we're going to talk about is DIYbio, and what exactly is it?
Yeah, so I'll start by just generally saying what we want to do with biology, which is, we want to know about health, drugs, disease. We want to know about the environment, trees, and other living things. We want to know how to grow food and how to make beer. And this is all like, yeah, beer. One guy, I don't know. And we want to know about longevity. That's like huge, we want to know how to live forever. And, that's sort of just an ongoing thing. And then I guess in terms of the theme of descent, if you think about the traditional training to become a biologist, you have to, blow 100 grand and get some kind of degree and take like 10 years until you actually get some kind of job where you can do your own work kind of thing. And it's sort of like a very patent-driven, closed, private industry. So right now it's a bit of like an all-boys club where you can't really get in unless you have all the money and time to put into this. And so there's a huge drive to take it out of the institution and start doing cool stuff now.
Yeah, for sure. So the good news is that if any of you have been around for the sort of the age when computing changed from super expensive, super exclusive, locked up in university labs, military labs, corporate labs where a computer cost $1 million, you had to be a PhD student to even get access to a computer. And then you look at, okay, well, what happened in the early 80s and definitely the 90s where computers became cheap, they went on your desktop. Early 90s, people started sharing code. You didn't have to write everything from scratch. And barrier to entry to get involved in computing, whether you are a user or a programmer, was lowered considerably. And right now, we're witnessing the same thing happening for biology. So as Kevin said, you might, before, maybe a few years ago, you might need a $100,000 degree, 10 years of your life, et cetera, et cetera. That's totally not the case right now. And it's really, really an exciting time for biology. And we'll just talk a little bit more about why it's so exciting.
Yeah, so the next cool analogy between, bio and computing is that, genes are code, and some people don't like this analogy, but whatever, it works. And so behind me you can see this coding device, which codes for something called a fluorescent protein, and this is a protein that comes from some jellyfish that somebody found, and it basically, is a sequence of letters, and when you put the DNA into your bacteria, it changes the color of it and makes it so that it glows red under UV light. So if you flip to the next, or you have something else? Right. So this is sort of what it looks like. And this is very much like the Hello World kind of experiment for DIY Bio. You transform your RFP and it's, oh my God, it glows, amazing. And it's like your LED board. So I think I have one more picture where you can literally just like paint pictures of, really cool stuff. Thank you, yeah. I didn't paint this, but it's cool anyways.
Yeah, and something that's cool, so if you can sort of play with us and go along with the idea that DNA is code, and if we actually do have the kind of tools today to be able to edit DNA code, I mean, DNA is just 4 letters, A, C, G, and T, just text, and so that means that with a computer you can manipulate DNA code, which is exciting. And so there are tools that are out there that allow people to do that. So before on this slide here, we had a tool that showed a coding device, and this is part of the BioBricks database. But there are an exploding number of tools that are generally made by open source community now that are available. So you don't have to pay $10,000 for a DNA design tool like an IDE. You can go online and take a look at a free one. So here's an example of one. It's like in a web browser. There's DNA code in here. It doesn't really mean too much to me to look at a glance, but if you can drill down and see these coding regions, which you can kind of think as functions, you can literally cut and paste those out as if you were cutting and pasting a function in a JavaScript program, and then paste that into a new DNA document. And with a bit of munging and thinking and learning, you'll actually be able to have that new organism express that piece of genetic code.
Yeah, so next, just to mention a bit of like the hardware that you need to start doing biohacking. So for one on the right, my right, your left, I guess, these are micropipetters, which are basically used to move small amounts of liquids between, from 1 microliter to 1 milliliter, typically. And you can get these on AliExpress for pretty cheap. And you can feel that they are pretty cheap when you get them, but at least it's, you know, inexpensive and you can use it for a little while. And on the right side, I guess you have these incubators, which you would use, you know, to incubate your bacteria at 37 degrees, and then they would basically grow into a culture overnight. And then you have your typical chemistry sort of glassware stuff.
And why would we want to incubate our bacteria overnight? What does that do?
Well, depends on what bacteria you're working with. I mean, yeast grows at 30 degrees Celsius, but the bacteria that is the lab strain E. coli, which is completely safe, comes from your gut, and your gut is at 37 degrees Celsius, which is your, you guys work in Fahrenheit, right? Something like that? I don't know. Sorry. Anyways, 37 degrees Celsius, which, yeah, is the temperature inside of your gut, and therefore that's the ideal temperature for these bacteria to grow. So that's how you get them to basically double in population in like 20 minutes. So
And so they grow exponentially, right? So
Exactly.
There's like this link of computing is an exponential technology. Biology is also an exponential technology. Is that
Yeah.
basically what you're saying?
yeah.
Cool. We have another tool here. So this is an example of a DIY PCI or PCR machine. PCR aids in the help of basically creating more DNA. So if you get a piece of DNA and you want to get more of it, you can do this process called polymerase chain reaction. And if you were to buy one of these products that comes from a big corporation, you can easily spend $10,000, $15,000, $20,000, depending on how cool of an item you want. This one costs about $1,000, and you can just buy it online, and it works pretty good. I've used it once, and it worked great.
So that's the Open PCR project, which is, I think you can build it yourself even too. The instructions are online somewhere. And then the one on the top left is called the Open Fuge, which is a centrifuge, also an essential tool in doing this kind of stuff, because after you grow your bacteria, it's sort of in a liquid, and you want to get the bacteria out of the liquid, so you just spin it really fast, and then all the bacteria get shot down to the bottom of the tube, and you can dump out the liquid, and they form like a dense pellet, basically. But yeah, and just to send a call out, these pipettes are one of the more annoying things and that haven't been hacked yet for doing bio. So if anyone feels like hacking a pipette and, you know, figuring out a way to move small amounts of liquids, then that'd be amazing.
That actually brings up an interesting point in that, you know, the biohacking scene is necessarily multidisciplinary. So, for example, in our experience in Brico Bio, we have biologists like Kevin who really know a lot about the biology. There's people like me who have a computing background, but there's also people that have engineering backgrounds, art backgrounds, law backgrounds. It's the kind of thing where, because it's so tactile compared to writing code, for example, it's really easy to get involved. So it's kind of like, you know, if you've been in a kitchen before, you're probably familiar with a good portion of the kind of things that happen in a biolab. Biolabs are just far more precise and have more expensive spoons and cups.
Yeah.
But you get to make cooler things. You get to make things that make themselves. So if I'm in a kitchen and I make a cake, I don't put the cake into an incubator and then wake up the next morning with 80 cakes. Which would be neat. But the cool thing about biology is that, you know, you're making these living products that can do work on your behalf. So the pictures that Kevin showed before, the work that those microorganisms were doing were creating colors. It was almost like you could say they're growing paint or they're growing ink or something like that. And if you think about the cost of printer ink by weight, I think it's something like the same price as gold or in the same kind of ballpark as something way more exactly. It's insane. And you know, with biohacking, you know, there's a real possibility that somebody could develop a product that allows you to grow your printer ink at home for, you know, very low cost, and it's totally sustainable. And there's people working on projects like this already, for example. And of course, that takes, you know, people that know the biology, people that know chemistry, and also maybe artistic people that have a deep understanding of color and what makes a good blue, for example, which don't ask me because, I don't know, I'm colorblind or something. But anyways, next slide. So we have something really cool here. So, you know, in the lab, you know, you do have to use these tools like pipettes and Bunsen burners and glassware. And to somebody like me versus Kevin, who is a real biologist who has had years of training in real labs in universities and companies, versus me, who I've only used DIY tools. And at that point, not very good. And one of the reasons why my background is in computing is because I can't hold a pen or a pencil and draw something on a piece of paper and have it look very good. I can type really well, but I'm not that kind of person that has that kind of dexterity. So I'm really poor. Perfect lab skills myself, and I can just download those lab skills.
Yeah.
And this is a real thing. So this is a project that's coming out of GenSpace and Tisch. It's called OpenTrons. It's less than a year old. You can well, they don't sell them anymore, but they used to sell these DIY kits, and they're working on something even better now.
Yeah.
But keep your eyes open for stuff like this. Not just OpenTrons, but there's other people that are working in this space. And it's kind of